It's a part 2 of amino acid and proteins lecture, and today's topic of discussion is peptide bond. As we know that all the amino acids are joined together by a bond, and ultimately they form a polypeptide. And after that, that polypeptide will undergo some changes like primary, secondary, tertiary, and quaternary structure, and ultimately it will form a functional protein. So it's very important that two amino acid will join together through a specific bond, which is covalent type of covalent bond, and which is known as peptide bond. On the first part, we have discussed about all the amino acid and its property, the type of amino acid like non-polar aliphatic R group, aromatic, polar and uncharged group, positive charge amino acid, and negatively charged amino acid. The next is peptide bond. Two amino acid molecules can be covalently joined through a substituted amide linkage term or peptide bond to yield a dipeptide. When there is a two amino acid, these two amino acids are together joined by a bond which is known as peptide bond. This peptide bond is a type of a covalent bond, and when this this two amino acid will form a bond together, they will form a dipeptide. Such linkage is formed by a removal of the element of water, dehydration, from the carboxylic group of one amino acid and the amino group of another. This bond, when this bond is formed, there is a elimination of a water molecules. So there will be a dehydration. So as we can see that this is a carboxylic group, and from the carboxylic group, this OH, and from the amino group, this H will be removed together, and they will form a water molecules. So after the removal of water, they will form this bond which is known as a peptide bond. Peptide bond formation is an example of condensation reaction. So when this process takes place and there is a removal of water and formation of this bond, this whole process is known as a condensation reaction. So as we can see in this chapter, there is a formation of peptide bond by condensation. The alpha amino group of one amino acid with RT group act as a nucleophile to displace the hydroxyl group of another amino acid with R1 group. So this RT group will act as a nucleophile, and it will replace this OH group from the amino acid 1. Okay, and forming a peptide bond which is shaded in yellow. This is a peptide bond. This is C, and there is a hydrogen, and on the carboxylic group there is a oxygen. Amino group are good nucleophiles, but the hydroxyl group is poor leaving group and is not really displayed. Okay, so as we can see that there is a displacement of hydrogen water molecules, and they will form this peptide bond. This peptide bond has some properties. Three amino acid can be joined by a two peptide bond to form a tripeptide. Similarly, amino acid can be linked to form a tetrapeptide, pentapeptide, and so forth. When a few amino acids are joined in this fashion, the structure is called an oligopeptide. So just like when two amino acids are joined together, they will form a when there is a two amino acid, okay, and when they will form, it is known as dipeptide. But when there is a three amino acid and they will join together by a two peptide bond, this is a peptide number 1 and this is a peptide number 2. So this is first amino acid, this is a second amino acid, and this is a three amino acid, and these three amino acids are joined together by a two peptide bond. It is known as tetrapeptide. Similarly, when there is a addition of one another peptide bond, it will form, and four amino acid, it is known as tetrapeptide, and then there is a pentapeptide, and so forth. So after that, some of amino acid will join together with the peptide bond, and they will form this oligopeptide. When amino acids are joined, the product is called polypeptide. When many amino acids are joined together, they will form a polypeptide. Protein may have 1,000 of amino acid residues. So when this chain is long, so when many amino acids are joined together, they will ultimately form a polypeptide, and this polypeptide will ultimately will form protein. Although the term protein and polypeptide are sometimes used interchangeably, molecules referred to as a polypeptide generally have a molecular weight 10,000, and those are called protein have a higher molecular weight. So we usually what we can do is like we call it polypeptide and protein both, but there is some difference. Like when we call it polypeptide, its molecular weight is like between less than 10,000. Okay, when its molecular weight is less than 10,000, it is called polypeptide. But this molecular weight is more than 10,000, then it will be called as a protein. So this is a major difference between how when we will call it polypeptide and when we will call it protein. So it totally depends on the molecular weight. When the weight is 10,000, it is called as polypeptide below the 10,000, and when it is above the 10,000, it will called as protein. So as we can see that this is a peptide bond, this is a peptide bond, this is a peptide bond, and this is a peptide bond. So these peptide bonds are together. Okay, so this peptide bond will together form a oligopeptide, then polypeptide. Okay, so the peptide are named the peptide seriglycerotyrosyl amino leucine are serine, glycine, tyrosine, alanine, leucine. Peptide are named beginning with the amino terminal residue, which by convection is placed at the left. The peptide bond are shaded in yellow. The R group are in red. Okay, so this is a R group. Okay, so as we know that all the amino acid have this chiral center, and through this chiral carbon there is a R group. Okay, this is a R group, this is a amino group, this is a carboxylic group, and this is a hydrogen, which is these all structure are same in all these structures are same in all the amino acid. Only this R group is different in different amino acid. Okay, so all these amino acids are joined together with the peptide bond. This is a peptide bond. So this is a peptide bond, and ultimately they will form a polypeptide, and after when its molecular weight is more than 10,000, then it will form a protein. The properties of peptide, the peptides, there are some properties of peptide which includes, the first is the polypeptides, yeah, or we call it peptide, has a partial double bond character. What we mean by partial double bond character, it is not a full double bond. Okay, it's not that it's a totally double bond character. No, it has a partial double bond character. So because of that partial double bond character, it will not rotate. So this occur due to the resonance, and because of that reason it will not rotate. Okay, so this is the amino acid A. Okay, and this is amino acid B. So they are together bond by a polypeptide, and it has this partial double bond character. It is not like this. Okay, it has this is a partial double bond character. The polypeptide backbone is relatively polar. They have a partial negative charge and partial positive charge because of the presence of carboxylic and amino group, but they are not ionizable. Okay, they are just polar group. Peptide bond absorb 4 UV light of 190 nanometer and 220 nanometer. Peptide bond is planar. They have a planar structure. There is a no rotation is allowed along the peptide bond. Okay, because of the partial double bond character. And this peptide bond is called as omega. When we read Ramachandran plot, then we will understand what is phi, psi, and omega. But for today's knowledge, we just need to understand that when these two together are joined, this bond is known as omega. And this is it has partial double bond character. It is polar, backbone is relatively polar. It absorbs UV light of 190 nanometer to 200 nanometer, and there is a no rotation allowed in this peptide bond. Generally, we have trans or cis peptide bond. All the amino acid except proline are a trans peptide bond. So except proline, it has a cis bond which is on its only terminal region. Okay, so on the terminal region, it will form a proline will form a cis, otherwise it will also form a trans. Okay, but other than this, all the amino acid will form only trans uh, peptide bond. So that's all about the properties of peptide. Thank you.